In lecture 9, we are going to be looking at biotechnology. We will be examining various techniques to clone a gene and insert that gene into a bacteria, and then we can use the bacteria's machinery to mass produce certain types of proteins that we want, such as insulin, for example. We are going to be looking at techniques such as DNA fingerprinting, and we are going to also look at gene therapy. So what is biotechnology? Biotechnology is basically exactly what it sounds like. It is technology that is based on biology. And in using biotechnology, we attempt to control, to alter, and harness cellular as well as biomolecular processes so that we can develop technologies and products to help improve our lives. Let's start with the concept of recombinant DNA. Recombinant DNA is basically a fragment of DNA that's composed of sequences originating from at least two different sources, so two different species, right? So an example here is we have a mice that has been incorporated with a protein called GFP, which stands for green fluorescent protein, and this GFP is actually taken from a jellyfish, and as a result the mice are going to be glowing in the dark. So you know, there are actually medicinal application of doing something like that. People don't just do it for amusement, and we're going to look at some examples of that later on. We can also use it, recombinant DNA, that is. We can also use recombinant DNA in agricultural application. So here is an example of where we use recombinant DNA for agricultural as well as sort of like a medicinal application. Okay, there is something called golden rice. Now, in some poorer countries, there is usually a lack of vitamin A in people's diet or in access to food that has sufficient vitamin A in them, and as a result of that, a lot of people suffer from vitamin A deficiency. And vitamin A is actually important for your vision. There is a type of cells in your eyes called the rod cells, and those rod cells are needed for nighttime vision. And with vitamin A deficiency, those cells don't quite work, and so it can lead to nighttime blindness. Okay, and in fact, in many poor countries, vitamin A deficiency is the leading cause of preventable childhood blindness. And also with the lack of vitamin A, it lowers people's immunity. So combine that with low vaccination rate seen in developing countries, that could actually help spark an endemic for various infectious diseases. So in some countries, each year it is estimated the vitamin A deficiency is estimated to kill 670,000 children under the age of 5 and cause an additional 500,000 cases of irreversible childhood blindness. Okay, so clearly this is something that's quite serious, and you know, if something can be done to help with the problem, then perhaps we should do it. So here is a map that shows you where the vitamin A deficiency is more severe across the globe, and you can see in developed countries like Canada, North America, right, Australia, there are virtually no significant cases, but in various developing countries there are quite a lot of cases of vitamin A deficiency. So that's where golden rice came in. Okay, so golden rice is a genetically engineered rice that is created to make beta carotene, okay, which is a building block for vitamin A. So beta carotene is not something that rice normally makes, and so in order for us to cause the rice to make the beta carotene, you need to insert genes from other sources, and so the golden rice was created by incorporating two beta carotene biosynthesis genes. These are genes that allows the creation, biosynthesis of beta carotene in the rice. So there was a Psy gene, PSY, from daffodils, the flowers, as well as another a gene called CRT1 from a type of soil bacterium. So by creating this recombinant genetics and giving it to the rice, scientists were able to produce rice that would now have beta carotene in them. So that should help solve the a problem of vitamin A deficiency if you're able to grow these golden rice in countries that needs it. But there were some controversial issues. Right? So an early issue was that the golden rice that was originally grown did not have sufficient amount of vitamin A, and so it was really not helping with the deficiency. So this problem was actually solved by development of a new strains of the rice. It was called golden rice 2.0, and that contains 23 times more beta carotene than the original version. Greenpeace actually opposes the use of any patent genetically modified organisms in agriculture, and they opposes the cultivation of golden rice because they think it would open the door to more widespread use of GMOs. Some people worry that there will be corporate control of agriculture in poor countries. Right? Typically, these are the countries where the golden rice will be grown, and if large corporations have complete control over how and where and what to grow in these poor countries, then many worries that that's going to lead to other type of ethical issues. Other argues that it is simply sufficient to get vitamin A from other food sources. So rather than creating a new GMOs for it, a genetically modified organism such as golden rice to solve the problem, maybe we should just get a food that has high vitamin A in them to these places. However, the counter argument for that is that those kind of foods are only typically available seasonally, and even if you're able to get those food to those countries, it will be too expensive for poor families to buy them. So golden rice, many argued, was a economically feasible option to treat the vitamin A deficiency problems. So that was an example of how we used recombinant DNA to create a new strain of rice to grow something, right? So it's kind of like an agricultural application, which then was used to fix a medical situation, the widespread vitamin A deficiency in some poor countries. Now let's take a look at another example. There is something called insulin. Insulin is it's a hormone that you make in the pancreas, and you would need insulin to bring your sugar level down. So after a meal, the pancreas will release the insulin, which would then tell your cells to absorb those glucose from your blood so you can use it for cellular respiration. The insulin will also tell the liver to absorb the glucose and store it as glycogen. So in people who have type 1 diabetes, their body fails to make the insulin. Okay, they don't have the insulin, and that results in very high blood sugar level all the time, and that has a lot of serious consequences as a result of that. So you have to give these people injections of insulin for them to have normal bodily function. So there were two scientists, Benting and Best. They're actually Canadians, and they were the ones who actually won the Nobel Prize for isolating insulin from animals, and knowing that was a compound that would bring the sugar level down. So prior to the late 20th century, the primary commercial sources of insulin were actually from cows and from pigs. Okay, so we would isolate the insulin from the calves and the pigs, and we would give them to people who have type 1 diabetes. And because these are animals' version of the insulin, they're not always effective, and you can't really achieve a very high purity as well. So they're not 100% effective as you want them to be, and so by late 20th century, using genetically engineered bacteria, there was something called humanin, okay, which is basically insulin for humans, and we were able to produce insulin at a much faster and larger scale. So how exactly does that work? Well, we would have to insert the insulin gene, which is not part of the bacteria. So this would also be an example of creating recombinant DNA. Okay, so we actually have 4 steps to do that. Okay, we have to locate a gene of interest first. We have to know where it is, right? And after finding it, the location of a gene, the locus of the gene, right, on a particular chromosome, for example, we would have to have a way to clone the DNA. Okay, we have to make many copies of the gene, and then we would have to insert it into a vector, and the vector is responsible for delivering it to, say, the bacteria, and then you basically have to just let nature do the rest. Okay? Once the bacteria receive the vector containing the gene of interest, in this case the insulin gene, then the bacteria machinery will start to make many insulin proteins from that gene that you have basically fed it, right? So that's what we're going to be looking at in the bulk of the lecture. We're going to look at how to do these 4 things. Step 1 is to locate a gene of interest, and that's actually a lot easier to do nowadays compared to, say, 2-3 decades ago, and that's because of the Human Genome Project. The Human Genome Project was an international and collaborative research program with the goal of mapping out the entire human genome. Okay, so they wanted to figure out the exact nucleotides that makes up all the chromosomes, and they want to figure out where the genes are located on these chromosomes and the functions of these genes. So it started on October 1st, 1990, and it took 13 years to complete the entire project. And with the completion of the Human Genome Project, it gave us the ability for the first time to read nature's complete genetic blueprint for building a human being. So there is something called a genome browser. All the results from the Human Genome Projects are basically accessible to public nowadays, and I'm going to show you how we can look for a particular gene of interest using the Human Genome Browser. So here it is, and you can get to this link by clicking on the image on the PowerPoint slide in case you want to play around with it. So right now we are on chromosome 17, and you can see this is the short arm of the chromosome, the P arm, and then we have the long arm here, called the Q arm, and the middle is where the centromere is, right? You might remember the centromere is where the 2 sister chromatids are going to be attached after DNA replication. So we are at this particular location on the Q arm of the chromosome, and here we can see there's a gene called BRCA1. That's actually the breast cancer gene 1. People who have a mutation in the BRCA1 gene are more likely to develop breast cancer. You might have heard of the movie star Angelina Jolie. She had a preventive mastectomy surgery because she actually had the mutation in the BRCA1 gene that could increase her risk of getting breast cancer later on in life, and that's why she decided to underwent the surgery, the double mastectomy. So we were talking about insulin earlier, so I'm gonna key in the insulin gene. I happen to know that it is INS, and there's going to be a bunch of a thing here, but this is the one I want, right? INS, insulin, decreases blood sugar concentration, right? It actually tells you what the function of the gene is. So if I click on that and I say go, we are taken to the insulin gene here, and we can see it is on chromosome 11, P15.5. That would be the locus of the gene, the location of the gene, right? And it is going to be 1,563 base pair long. If I click into the gene, we will be taken to a more detailed page, and you can look at various information about this gene, right? For example, the protein structure, the insulin structure, right? We can take a look at what that looks like. So here, that's the ribbon structure of the protein, right? You can see there are some alpha helices there, and this is another view of the protein. We can click on the genomic sequence, and you have various options, right? You can turn on the introns, turn off the introns, depending what you want. If you want to insert this into a bacteria, you can probably remove the introns because they are non-coding anyways. And if you submit it, then that's the entire sequence for your insulin gene. Okay, so this is the instruction that would allow the body to make the insulin. So if this was in a human cell, then you would make an RNA transcript from this, and then in the cytoplasm, the ribosome would decode the mRNA transcript to make the protein. Right? And if I go back here, I can also look at the protein. Right? It's a relatively short polypeptide. It's only 110 amino acids, and they're all listed here. So each of these actually stands for a particular amino acid, right? M is methionine. We know the first one has to be methionine, right? Because the start codon is AUG, and that also codes for methionine. So that's why we have M always. Then next one is alanine. Next one is leucine, and so on and so forth. And this is essentially the insulin sequence. Okay, so you can see it's very convenient with the genome browser. You can look up any gene as long as you know the name of the protein or of the gene that you're looking for. You can find out exactly where it is in the human genome. Now that we have located the gene of interest, now that we know where the gene is on the chromosomes in the genome, we have to clone it. We have to make multiple copies of it. So prior to the late 1980s, in order to clone a gene, in order to clone a segment of DNA, you would have to feed it to a bacteria. You would have to let the bacteria divide, and every time the bacteria divide, it would kind of make a copy of the DNA that you've inserted into them. And after multiple cycle of bacterial division, you would have to harness the DNA from the bacteria again. So it was a very tedious work, very labor intensive, and it takes quite a bit of time before you can clone the DNA. But in the late 1900s, there was an innovative technique called PCR. So PCR stands for polymerase chain reaction. This technique allows you to take a specific DNA segment and you can copy it many times without ever putting it into any cell. Okay, and you can actually do it in a very short period of time. It literally just take a couple hours, and you would end up having tons of copies of the original DNA. Here is what a PCR machine looks like. This is kind of where you would load your sample, the ones that you would want to clone, and you would program the machine and set the different temperatures to help clone the DNA. We're going to get into the temperature parameters in the next slide. But the good thing about this is you can actually use it on any sample of DNA with at least just one copy of the sample. So this could be a DNA sample from a sperm. It could be a DNA sample from blood that is taken from a crime scene. It could be remnants DNA from say a mummy or something. Right? You just need a very small amount of the DNA, one molecule of DNA, and then you put it in the PCR machine. And each time the cycle runs is going to double that number. So if you do the math, right? If you do it in 10 cycles, to the exponent 10 is 1,024 copies. And if you run it for 30 cycles, right? 30 cycles, and I cannot do that in my head, but if you were to plug it in a calculator, it would tell you that there is going to be billions of copies, right? And so you can do this under several hours. Okay, so you start with one copy and you end up having billions of copies. Very convenient, very easy to use. The methodology of PCR is actually closely related to the process of DNA replication, and that's something that we've learned earlier in the course. We know that in a cell, the DNA strands are going to be separated using gyrase and helicase. Right, the helicase is the one that will unwind the DNA, and the gyrase is the one that's going to be relieving the tension in the DNA as you unwind. So instead of using the helicase to unwind the DNA, we are actually going to be separating the DNA strand with high temperature. Okay, so when you heat up the DNA, because the two strands of the DNA are essentially held together by hydrogen bonds, they are relatively weak bonds that can be broken when the temperature gets high. So we would use that to separate the strand. So that takes away the need to have helicase. And because whatever you're cloning is not too long, so there is not going to be enough tension that will build up and break the DNA or anything like that. So gyrase would also not be necessary. And once the strands are separated, each strand can be used as a template to build the complementary strand, just like DNA replication. So in PCR, we have to create what's called DNA primer to kind of mark the location, the region that we want to clone. Okay, when we have a DNA sample, we have to have an idea of what's flanking the target section. Okay, so if this is the region that I want to make multiple copies of, then I have to design a primer that would be upstream of that section as well as downstream of the section. Okay, you remember the primer in DNA replication, right? There is the primase, the enzyme that will put down an RNA primer, and that will guide the polymerase to the site of replication, where they will start to build the new strands from 5' to 3'. So instead of using an RNA primer, we will be using a DNA primer, so we don't have to replace them later. And so you have to choose the primer such that it will flank your sequence of interest. Instead of using DNA polymerase 3, we will actually be using something called tag polymerase. Tag polymerase is actually polymerase that has been isolated from hot spring bacteria. So these bacteria live in really high temperature environment, which is suitable for the PCR reaction, because after all, we are heating it up to around 95 degrees Celsius at some point of the PCR process. So if we were to use the regular DNA polymerase, that would be denatured and it will no longer be functional. But not with the tag polymerase. It is used to high temperature, so that's not going to be a problem at all. We have the original DNA strand here, the one that we're trying to copy. So you would separate them using heat it up to about 95 degrees. And after the strands separate, you would lower the temperature a little bit to allow the primers to bind. And then the tag polymerase will know where to start, and they will continue to build on the 3' end. So one tag polymerase will build this way, and then the other will build this way. And afterwards, you would have 2 copies. And if you let this go again in another cycle, then you would have 2 times 2, resulting in 4 copies, and then 8 copies, and then 16 copies, and so on and so forth. So here we can see just how the cloning process work. And again, we start with the original DNA sequence, followed by step 1, denaturation. Okay, so in denaturation, you are going to heat up the solution. So the PCR machine will bring the temperature up to around 94 degrees to 96 degrees. That will separate the DNA strands, and then afterwards you would lower the temperature a little bit to allow the primers to bind to it. Okay, so when you bring it down to about 68 degrees, right, the primers will bind, and then you will bring up the temperature a little bit to 72, just enough for the tag polymerase to do its work. Because if the temperature is too low, then the tag polymerase doesn't know what's going on, right? Remember, these are polymerase that are taken from hot spring bacteria, so they're used to high temperature. So if it's too low of temperature, they won't be functional. So by heating it up a little bit, then they are going to be able to build the new strands. So you end up having 2 copies, and then you just repeat step 1, 2, 3 again, and then you will have 4 copies, and so on and so forth. So that's basically how PCR works. The exact temperatures might have to manipulate it depending on the gene that you're cloning. Sometimes if it's rich in C's and G's, then you might need to have a higher denaturation temperature. If it's rich in A and T's, then you might need a lower denaturation temperature. because there are only 2 hydrogen bonds holding A and T's together, but 3 hydrogen bonds holding C and G's together. So it would be more difficult to break something that has more C's and G's in them. But we don't really have to worry about it until you actually do a PCR reactions. If you do that sometime in your life, then you will learn to play around with these temperatures. But the key point here is PCR is a way to clone a short segment of DNA, and we can do it in very short period of time. So in our journey to create the recombinant DNA to kind of hijack the bacteria's machinery to mass produce a particular protein, we located the gene of interest, the insulin in this case. We cloned the insulin gene using PCR. Then now it's time to insert that DNA into a vector. And I think I'm actually going to do that in the next video instead. So thanks for watching.